Vanakkam. Welcome to this course on classics in neuroscience. In this video, in this series of videos, we have been discussing cell biology and the description of the synapse. In this video, we will be discussing the electrical synapse. So far, we have been discussing the chemical synapse, and in this video, we will be discussing the electrical synapse. We will discuss how the electrical synapse essentially presented a relatively new perspective of the neuron doctrine, and the contribution of Betty Guerin and myelin, the nature of myelin. Soon after the demonstration of the chemical synapse, electrical interaction between neuronal cells were demonstrated, first reported by the lab of Bernard Katz. Remember, Bernard Katz is the person who first studied, or one of the pioneer of chemical transmission in neuromuscular junction, who described the neuromuscular communication through vesicles. This Edward Fushpan and David Potter were postdocs at NSF. They investigated an invertebrate model. They studied the nerve cord of crayfish. And what they found was that there was simultaneous action potential generated in the presynaptic and the postsynaptic neurons. So, the specific case they studied was a neuronal axon connecting to a large motor axon, essentially an axonic synapse. Remember, this is important. Usually communication happens between an axon and a dendrite, or between an axon and, in the case of neuromuscular junction, a muscle fiber, for example. In this case, this communication that Fushpan and Potter studied was between an axon and another axon. So, Fushpan and Potter used microelectrodes, a pioneering instrument or a pioneering equipment at that time, that is the intracellular microelectrode. That is the intracellular, that is the microelectrode that could be placed inside the cell without damaging the cell, intracellular microelectrode. So, when they used an intracellular microelectrode to stimulate this axon so much, such that this axon produces an action potential, they found that a similar action potential was found in the postsynaptic neuron. But that is not surprising, because if there is an action potential in the presynaptic neuron, there will be an action potential in the postsynaptic neuron. However, the synaptic delay was very minimal, suggesting that there was practically very minimal or no chemical transmission, that this should have happened through mechanisms of action potential itself, not through chemical messengers, was the suggestion. But more importantly, what they also found was that if they use graded potentials, that is potentials that were too small to produce an action potential in the presynaptic neuron, this will not produce an action potential in the presynaptic neuron. So, essentially vesicles containing neurotransmitters will not be released into the synaptic cleft, and so action potential will not be there, action potential or any potential will not be there in the postsynaptic neuron. This is called graded potentials. We will provide you with a brief video of explaining what is an action potential and what is a graded potential. It will be shared in the forums. So, a potential that is too small to produce an action potential or too weak to cause an action potential is called as a graded potential. When they stimulated the presynaptic neuron with a graded potential, they found that the postsynaptic neuron also had a graded potential. This is convincing evidence, because if there was chemical transmission, it can only happen when an action potential happens because it is the action potential that causes release of the neurotransmitter, and it causes an action potential in the postsynaptic neuron. But a graded potential cannot transmit itself. A graded potential is something that does not cause any effect on the postsynaptic neuron. In this case, they also found an equivalent weak response when a graded potential was induced in the presynaptic neuron. This convincingly suggested that there was direct current passage between one neuron and another neuron. Type of connection in which neurons communicate electrically is called as the electrical synapse. However, suppose you stimulate the postsynaptic neuron, will it cause an action potential in the presynaptic neuron? The answer is no, because there was one-way transmission. So, communication was happening in one direction but not the other direction, as form of similar to rectification, like you have in full wave rectifier. It was initially believed that this was probably found only in simpler systems such as invertebrates, but now it has been suggested that this is also found in many other places. For example, in vertebrate brainstem, one place where this is discussed is the inferior olive, that also houses the climbing fibers that supply powerful signals to cerebellum. Another place that they are found in excitable cells, not necessarily in neurons, is the heart muscle, for example. In the meanwhile, there was widespread search to identify a structural foundation or structural basis for explaining this electrical synapse, because there must be some way in which communication must be enabled between these two different cells. These are two different cells, axon of one cell connecting with axon of another cell, but communication is happening. What is the medium through which it is happening? It was described by Revel and Karnowski in 1967 that there were gap junctions, very critical concept. Do Google this, gap junctions, that enable action potential communication from one to other, either in both directions or only in one direction. In this case, we are showing gap junctions that conduct electricity in both directions. What is in this picture, that is what is shown. They not only facilitate electricity conduction, but they also facilitate exchange of nutrients, exchange of other molecules, and molecular differentiation and between cells. So, a whole lot of molecules are exchanged between cells through the gap junctions. So, cell differentiation, nutrient exchange, growth, signal transfer, and a whole bunch of functions. And this is found in various parts of the body, especially the heart, playing roles in synchronous integration of brain and other functions. So, a critical concept. Do Google this, beyond the scope of this course, do check this. Now, a question is, now that we have established neuronal communication through electrical synapse and there are gap junctions between cells, is it that there is reticularism as claimed by Golgi and not neuron doctrine as suggested by Ramon Cajal? Is it that? So, see, the cells that are communicating, the neurons that are communicating still are individual cells with their own nucleus and with their own organelles, just that they are communicating directly with each other through exchange of molecules, but also through communication through electrical channels. So, gap junctions, the presence of gap junctions or electrical synapses do not actually invalidate the neuron doctrine, because cells, they maintain their own individuality and their own functional autonomy, suggesting that gap junctions and electrical synapses essentially add value to the neuron doctrine by revealing one more layer of complexity in the neuronal communication and interaction. This suggests that there is a coexistence of chemical synapses with electrical synapses in forming neural circuits, and gap junctions, they contribute to dynamic and intricate neural networks, suggesting that how complex our nervous system is. Essentially, this increases the understanding or increases the complexity of the neuron doctrine. It does not disprove or invalidate the neuron doctrine. It is not that it is the same neuron that is continuing. These are two different neurons that are communicating through electrical means. That is the point, that these are indeed two different neurons. So, the neuron doctrine is true, but there is a complexity. In 1954, Betty Geron discovered the existence of myelin and peripheral myelin formation. This discovery was at the Harvard University. So, she described myelin as a multi-layered wrapping by Schwann cells around the axon. What is myelin? Myelin is this fatty lipidy substance that surrounds large neurons. Myelin has been described since the 1800s, the late 1800s, but fine structural description of the myelin happened only in the mid 20th century or the 1950s with the advent of the electron microscope. Immediate confirmation of myelin was done by Geron and Schmidt in 1954 and Robertson in 1955. It turns out that myelin plays a critical role in improving or in increasing the resistance, that the membrane resistance, and reducing the capacitance, thus aiding rapid conduct of action potential. So, if there is more and more myelin, the conduct of action potential through that axon that is having more myelin will be faster. Why is this the case? I will share with you a video where we discuss how myelination interacts with conduct and the speed of conduct and the length of conduct, with two different concepts called length constant and time constant. We have discussed this in a previous course. My teaching assistants will share with you this via forums and maybe in YouTube description also. So, also when myelination disappears or for whatever reason, if myelination is affected, it leads to disorders such as multiple sclerosis and Guillain-Barré syndrome. We will also share a video that describes these. These are outside this course. Those who are interested can check how myelination improves the speed of conduction. Physiology, but still there are principles of engineering, electricity, physics that are applied here, that still work here. Do check out those extra videos. So, these are the references that we use for the past few videos, the important papers. So, in this video we looked at the electrical synapse, the nature of electrical synapse, especially as described by Fushpan and Potter in an axo-axonic synapse, producing both an action potential and also a graded potential. And does it mean that the neuron doctrine is invalidated? No, it just means that the neuron doctrine has become more complicated. And then description of myelin by Betty Geron and how myelin, or what is myelin and its role in increasing the speed of conduction in neurons, in myelinated neurons. With this we come to the end of this video. Thank you very much for your attention.